Okay, let's jump right in. The truly mind-blowing thing from this 2012 science paper right at the start.
Yeah.
It's this. One single protein, Cas9, right,
Okay.
guided by just two small RNA molecules.
Okay.
It can be programmed to cut pretty much any DNA sequence you want.
Any sequence, that's the kicker.
Exactly. One protein, these RNA guides, like molecular scissors you can program.
It totally changed how everyone thought about gene editing. A real game changer.
Before this paper, I mean, CRISPR was known, but more as this bacterial immune thing, right?
And it's right. It was understood as how bacteria defend themselves against viruses.
It'd store little bits of viral DNA.
Yeah, like molecular mugshots. Then they'd make matching RNAs, these cRNAs, to spot the virus if it showed up again.
But the big question mark was how that spotting led to actually destroying the virus's DNA.
Precisely. Which protein did the cutting? How was it guided? That was the mystery this paper really solved.
And the Doudna and Charpentier labs, they really zeroed in on this type 2 CRISPR system.
They did. And they fingered Cas9 as the protein doing the heavy lifting, the actual cutting.
Right. And figure 1, that's where they showed Cas9 isn't acting alone. It needs help.
Yeah, it needs those two RNA guides. That was a key finding right there.
So not just the cRNA that matches the target.
No, it also needs this other one called tracrRNA. You can think of the tracrRNA as sort of helping Cas9 hold the cRNA correctly.
Okay.
So in the experiment, what they did was mix purified Cas9 protein with a specific cRNA.
cRNA SP2, I think it was.
That's the one. And then they added plasmid DNA that had the matching sequence. Critically, it also had this little adjacent sequence, the PAM site.
Ah, yes, the PAM. We'll get back to that.
Definitely. But the key thing here was testing conditions. Cas9 cRNA, and then either with or without that tracrRNA.
And the result was pretty clear-cut, wasn't it?
Absolutely. You look at the gel in Figure 1A. The DNA plasmid only got cut when both the cRNA and the tracrRNA were there.
So Cas9 is this dual-RNA-guided DNA cutter, and endonuclease needs both guides.
Exactly. And it's sequence-specific.
Yep.
They used a control cRNA, SP1, that didn't match the target.
And
no cutting.
Yeah.
As you'd expect. Shows it's not just cutting randomly.
They also checked if the DNA shape mattered, using circular versus linear DNA.
Right. Figure 1A shows results for both supercoiled, circular, and linearized plasmids. The requirement for both RNAs held true.
Okay. And then Figure 1B, they looked closer at the cut itself, using radioactivity.
Yeah, they wanted to see if it was cutting one strand or both. So they labeled either the target strand or the non-target strand with a radioactive marker.
Clever. So you can track each strand separately.
Precisely. And the results showed fragments appearing for both labeled strands.
Meaning it cuts right through the double helix, a double strand break.
Correct. Not just a nick. And Figure 1C pins down exactly where those cuts happen.
How did they do that? Primer extension?
Yeah, primer extension termination sequencing. It's like tracing the DNA sequence with a copier, but the copier stops when it hits the break Cas9 made.
So where it stops tells you the exact cutting site.
Yes.
Exactly. To the nucleotide.
And Figure 1E ties it all together visually. Shows the cRNA pairing up with the tracrRNA.
That's the orange part in their schematic.
And also pairing with the target DNA, the protospacer.
The yellow part. And you see the gray PAM sequence sitting right next to that target region.
And the little arrows showing the cut sites mapped from Figure 1C.
Blue and red arrows, yeah. Shows precisely where Cas9 snips each strand.
Okay, so Cas9 plus 2 RNAs, it targeted DNA cutting. Now, Figure 2 digs into how Cas9 actually does the cutting, right? The domains.
Exactly. Proteins often have these modular parts, domains, that do specific jobs. Cas9 looked like it had two potential nucleus domains parts that cut DNA.
Based on sequence similarity to other known nucleases.
Right. One called HNH and another called RuvC-like. The hypothesis was maybe these were the molecular scissors.
So how'd they test that? Did they, break those parts?
Pretty much. They introduced specific mutations into the gene coding for Cas9, designed to knock out the function of the HNH domain in one version and the RuvC-like domain in another. So disable one potential scissor blade at a time.
You got it. Then they tested these mutant Cas9 proteins in the same DNA cutting assays as before with the cRNA and tracrRNA.
And what happened?
Really elegant results, actually. Look at Figure 2. When the HNH domain was mutated, Cas9 could still cut the non-complementary strand, but it lost the ability to cut the complementary strand, the one the cRNA directly binds to.
Okay, so HNH cuts the strand that matches the RNA guide.
Seems like it. And conversely, when the RuvC-like domain was mutated...
Let me guess, it cut the complementary strand, but not the other.
Exactly. It lost the ability to cut the non-complementary strand.
Wow, so that proves it. Two separate cutting sites within the Cas9 protein.
Yep. Each domain nicks one specific strand of the DNA double helix. They confirm this using both the plasmid assays and the labeled single-stranded DNA substrates, just like in Figure 1.
Really neat dissection of the protein's machinery. Okay, moving on to Figure 3, this one seems to be about the RNAs again, figuring out which bits are crucial.
That's right. Now that they knew Cas9 needed cRRNA and tracrRNA and how Cas9 itself cut, they wanted to know what are the minimal pieces of these RNAs that are required.
Makes sense. Did they just start chopping parts off?
Essentially, yes. Figures 3a and 3b show experiments using truncated versions, shortened versions of both tracrRNA and cRNA.
And they tested if these shorter RNAs could still guide Cas9 to cut DNA.
Right. And through this systematic shortening, they mapped out the essential core regions. Figure 3c gives us anectylin. The blue box highlights these minimal regions. They even identified what they called an activating domain within the tracrRNA that seemed crucial.
Interesting. And Figure 3 also introduced this idea of a seed sequence in the cRNA. What's that about?
Yes, the seed sequence. This is really important for understanding Cas9's specificity. How does it make sure it cuts the right target?
Okay.
They experimented by making tiny changes, single-base mutations in the target DNA sequence, specifically in the region where the cRNA binds.
And they looked at how those mutations affected cutting.
Exactly, both in the test tube, in vitro, and also in living bacteria, as pyogenes, using something called the transformation assay. What they found was striking.
What was it?
Mutations in the part of the target DNA that's closest to that pan sequence that had a huge effect.
The part the cRNA binds to first, maybe?
That's the idea. They call this the seed region. Even a single mismatch there could drastically reduce cutting or stop it altogether. You see that in the transformation efficiency shown in Figure 3D.
So the seed region is like the most critical part of the address Cas9 is looking for. Get that wrong and it doesn't work.
That's a good way to put it. It acts as the primary checkpoint for target recognition.
Figure 3e looks like it explored this more with mismatches between the cRNA and the target DNA itself.
Yes, they introduced mismatches systematically along the cRNA DNA binding interface. And again, mismatches within that seed sequence, the 812 nucleotides right next to the PAM, were the most detrimental to Cas9 activity.
Really highlights the importance of that specific region for accuracy.
Absolutely.
Yeah.
It ensures Cas9 doesn't just cut any sequence that's vaguely similar.
Okay, we keep mentioning this PAM sequence. Figure 4 finally puts it under the microscope. What's the big deal with the PAM?
The PAM protospacer adjacent motif. It's this short DNA sequence, just a few bases long.
NG in this case, right, for Streptococcus pyogenes Cas9.
Correct. NG, where N can be any base. This sequence must be present in the target DNA, right next to the sequence the cRNA binds to. It's non-negotiable.
So even if the cRNA matches the DNA perfectly, if that NG isn't there.
No cutting, or at least very, very inefficient cutting. Figure 4a shows this clearly. They tested target plasmids with mutations in the PAM sequence. Perfect cRNA match, but mutated PAM. Cas9 was basically inactive.
Wow, so it's not just the RNA guide. The DNA target itself needs this little tag.
Exactly. Think of it like the cRNA guides Cas9 to the right street address, but the PAM is the specific house number it needs to see before it'll act.
And Figure 4b shows this over time.
Yeah, it's a time course using labeled DNA. With the correct PAM, the WT or wild type, you see the DNA getting cut into smaller fragments pretty quickly. But with mutated PAMs, look at those lames. Basically nothing happens, even after an hour. Cutting is abolished.
So the PAM is essential for the cutting step, but Figure 4c suggests it's important even earlier, for binding.
Right. This is using a dead Cas9, a version mutated so it can bind DNA but can't cut it. They used an EM assay, an electrophoretic mobility shift assay.
That lets you see if proteins are binding to DNA because the complex moves slower through a gel.
Precisely. And what they saw was that this dead Cas9 could bind strongly to the target DNA only when the correct PAM was present.
So if the PAM is wrong,
binding is drastically reduced or gone altogether. So the PAM isn't just needed for cleavage. It's required for Cas9 to even properly lock onto the DNA target in the first place.
okay, that clarifies the PAM's role. So Cas9, cRNA, TracrRNA, PAM, that's the whole set of requirements.
That's the natural system they dissected.
Now, Figure 5. This feels like the moment where it transitions from understanding a bacterial system to creating a powerful tool, the single guide RNA.
Absolutely. This was a huge conceptual leap and a brilliant piece of engineering. They thought, okay, the cRNA and tracrRNA have to work together. They interact.
Right.
What if we just fuse them? connect the important part of the cRNA to the important part of the tracrRNA with a little linker loop.
Create one chimeric RNA molecule.
Exactly. A single guide RNA, or sgRNA. Figure 5a shows the design concept, linking the 3-foot end of the cRNA to the 5-foot end of the tracrRNA, often using a specific loop structure like a GAA tetraloop.
The big question then, would this artificial sgRNA work? Could it still guide Cas9?
That was the million-dollar question, and figure 5b shows the answer was yes.
It worked.
It worked beautifully.
Yeah.
Cas9 programmed with this single sgRNA cut the target DNA just as well as Cas9 with the 2 separate natural RNAs. They confirmed this using restriction enzymes. The cutting pattern showed the sgRNA-guided cleavage was happening correctly.
That's amazing. That simplification must have been key for making it practical.
Oh, massively. Suddenly, instead of needing 2 RNA components, you just needed one easy-to-design, easy-to-make sgRNA.
And they didn't just show it worked for one target. Figure 5D really drives home the programmability, right?
Yeah.
Targeting GFP.
Right. GFP, the green fluorescent protein, is a common lab tool. They designed 5 different sgRNAs, each programmed to target a different sequence within the GFP gene.
Yeah.
And they showed that each sgRNA directed Cas9 to cut specifically at its intended target site within the GFP plasmid.
So just by changing that 20 nucleotide guide sequence in the sgRNA,
You could redirect Cas9 to a completely new location on the DNA. That demonstrated the incredible programmability and potential for targeted editing anywhere in a genome.
Okay, let's pull back a bit. Thinking about the whole paper, what were the real landmark innovations here?
Well, number one, as the authors pointed out, was just achieving the in vitro reconstitution. Taking these components out of the bacteria and showing, yep, Cas9, cRNA, and tracrRNA work together outside the cell to cut DNA. That was fundamental.
Understanding the basic mechanism.
Right. Then second, the functional dissection of Cas9 itself, identifying those HNH and RuvC-like domains and proving they each cut one DNA strand. That gave crucial insight into how it worked.
Knowing your tool.
Exactly. Third, and maybe the most impactful from a practical standpoint, was the sgRNA concept, fusing the 2 RNAs into one. That was the stroke of genius that made CRISPR-Cas9 so accessible.
Yeah, the simplification factor.
Huge. And finally, number 4 was clearly demonstrating the programmability, showing you could target different sites, like those in the GFP gene, just by swapping out that short guide sequence in the sgRNA.
That really unlocked the potential for genome editing.
It really did. And it's interesting, they used pretty standard biochemical methods. The real breakthrough was the conceptual leap disassembling this natural immune system and then reassembling it into a simplified modular programmable tool.
And the collaboration between the labs was key too, bringing different expertise together?
Definitely. RNA biology, bacterial genetics, that cross-disciplinary vision was vital. They had the insight to test Cas9 outside its natural context and see the potential beyond just bacterial defense.
The paper itself even hints at the future, talking about developing RNA-programmable genome editing platforms.
They saw it coming. It really did lay the foundation. It shifted. The paradigm CRISPR wasn't just bacterial immunity anymore. It was a programmable nuclease.
And the speed at which other labs adopted it, the technical transferability must have been high because of that sgRNA simplification.
Absolutely. The citation count on this paper is just astronomical, which speaks volumes about its impact across all of science, medicine, agriculture, you name it.
It's a fantastic example of, like you said, old tools maybe, but a completely new way of thinking, a theoretical breakthrough.
Driven by that interdisciplinary approach and aiming for something with huge potential impact.
So thinking about that programmability again, the fact that it all boils down to simple RNA-DNA base pairing to target Cas9 really makes you wonder, doesn't it?
How so?
Well, what other biological processes rely on specific DNA interactions? Could we use similar programmable RNA-guided systems to influence those? Maybe not cutting, but recruiting other factors, turning genes on or off.
That's a really interesting thought. If the targeting is that straightforward and programmable,
Right.
what else could you potentially guide to specific DNA locations using RNA?
Exactly. It feels like the potential applications of this kind of RNA-guided programmability might extend far beyond just Cas9 and cutting DNA. Something for you, the listener, to think about.